together with my teammates Amir and Sadia, we are going to take you through our bioengineering project where we are looking to create an osmotic pressure-driven drug delivery system with the added functionality of pH sensitivity. Now, we may know that some of the most common diseases that we experience have to do with the gastric acidity, either hypersecretion or too low secretion of acid. For example, we have gastric reflux, which is a consequence of gastric hyperacidity. And on the other hand, we have indigestion or dyspepsia, which is a consequence of too low gastric acidity and high stomach pH. And these conditions actually have clinical and humanistic ramifications for their patients. And it so happens that pH-sensitive oral drug delivery is over-reliant at the moment on chemical compatibility between drug molecules and the pH-sensitive polymers. So to circumvent that, our project is to sort of incorporate pH-sensitive hydrogel in principle into an osmotic pressure-driven drug delivery device, which for the purpose of this project, we will call the microfluidic device, or the MFD. So we'll take you through our design, the materials we intend to use, the fabrication method, the assembly, and the characterization method, and our expected outcomes and potential implications of our projects.
So this is the design for our MFD. It's a very simple representation of what our final product will look like. We have two chambers over here, one containing the osmotic agents and one which are loaded with the drugs. These two agents are here separated by our push layer. We also have a semi-permeable membrane which surrounds the two chambers, which allows the movement of water. And finally, over here we have our orifice, which is then covered by our pH-sensitive hydrogel cap. How this works is that the osmotic agents pull in water from the environment, which then expands the first chamber and the push layer, pushing on the second chamber. If the pH conditions are right, the hydrogel cap will expand, opening the orifice, allowing the drug to move through the orifice and into our environment in a very controlled and precise manner. Once the pH is back to normal, the hydrogel cap will contract, closing the orifice, ensuring that we do not overdose our patients. Overall, this is a very simple design. The design of our MFD is very versatile and can be used for multiple different purposes. By making small changes in our design, we can change the way it functions. For example, by changing the material of the hydrogel cap, we can change its stimulus response. Moreover, to change the rate of osmosis, we can change the permeability and the dimensions of the semi-permeable membrane. We can also change the rate at which the drug is released by changing the size of our orifice and the properties of our push layer.
So we are going to continue with the materials of the components of the and go through their features and why we chose these materials. So the first one is the outer layer. So the outer layer should be high acidity resistance and semi-permeable membrane. The material we chose for this is the cellulose acetate, and because of the features of it, which is controlled water diffusion, high strength and flexibility, good thermal stability, semi-permeable membrane, and non-absorbed orally and easily exerted. The second component we are going to go through is the osmotic agent. So we're going to use both sucrose and fructose material for this part. So the reason is that they have high osmotic pressure, which is near to 430 atmosphere. And they are digestible carbohydrates, and they are orally non-toxic. So the third component we are going to go through is the pH-sensitive hydrogel cap. So it is made of two different hydrogels for two applications. So the first one is it is for high pH responsive acidic area, which is made of CMCPA, and for low pH responsive area, which it is made of CCPVP. So last but not least, we are going to go through the push layer. So it brings the two chambers together, the osmotic agents and the drug. So we are going to use HPMC because of its unique features of because it has it is solubility and swelling behavior, its controlled matrix formation, and osmotic driving force. So this was about the material part. So we're going to go for fabrication process. So, as we said, we have two chambers. This is the first chamber. This is the second one. So the first chamber is contained of the drug, which you compress it using granulation process, as well for the second chamber, which is contained of sucrose and fructose. So after we compress them, we bring them together, and we spray HPMC through them, and then we compress them together, and then we cellulose. We coat it using cellulose, and after that we laser drill to fill our orifice, and then we use sort of this in terms of cap. And as I said, we use 2 different hydrogels for 2 different responsive environments.
Okay, so we've gone through the design and fabrication of the MFD. So at the end of the day, we want to characterize it through a rigorous in vitro process to ensure that our design meets important quality parameters. And for that purpose, we'll be doing 4 major types of characterization processes. First, we'll do a chemical fingerprinting. So we'll be using techniques like FTIR, Fourier transform infrared spectroscopy, and proton NMR to obtain the chemical fingerprints of the functional groups of these different materials individually. After that, we will be doing a morphological and material properties characterization of the components again separately, and the MFD after it has been assembled. Thirdly, we'll be doing a drug loading and release characterization. For drug loading, essentially we want to calculate what percentage of the entire MFD by mass is occupied by the drug, and we would go through the fabrication and design processes in a holistic iterative process until we are able to optimize the encapsulation efficiency. For drug release, we are going to do a drug release characterization in the USP dissolution apparatus using simulated gastric fluid, and for this purpose we would immerse our microfluidic device in the apparatus and we draw sample aliquots at specific time points, and then we will use UV visible spectral photometric quantification to measure how much of the drug has been released into the simulated gastric fluid solution at this specific time point. We would also conduct statistical analysis to confirm that our device actually releases the drug following a zero order release kinetics. So we will also do a pH response and self-regulation test, and for this we will load the MFD with sodium bicarbonate instead of the antacid drug, and then we will immerse it in the dissolution apparatus again with using simulated gastric fluid. We draw sample aliquots at specific time points to concurrently quantify sodium ion concentration as well as the pH to compute the pH response lag as well as the self-response time to understand the response, the pH response of the system, and to sort of quantify our stimulus responsivity and compare it with literature findings of similar systems. Lastly, we are going to conduct an in vitro pharmacological test to study the efficacy, the biological efficacy of our drug-loaded MFD, and for this we'll be using the in vitro artificial stomach, a pharmacological model of gastric diseases that has been reported in literature. We are going to simulate fast acid release, slow acid release, as well as gradual pH increase in this in vitro system. Because this model is very robust and it is complex enough to recapitulate the physiological conditions, we consider it an appropriate alternative to in vivo models that of course have ethical issues. At the end of the day, using all these techniques, we want to holistically provide quantitative and qualitative data on the MFD and demonstrate the versatility of the system for stimulus responsive controlled drug release in an oral delivery scenario. So at the end of the project, we want to ensure that we have designed and fabricated a suitable microfluidic device that achieves zero order release kinetics of our drug of choice in a stimulus responsive manner that has also undergone rigorous testing and meets standard quality parameters as reported in literature. Along the line, we intend to have our work packages to design the fabrication and characterization in a whole iterative process, and we envisage that we might experience some challenges. For example, it's possible that the microfluidic device will not have adequate transit time and it will be elicted from the gastric fluid. If we do experience that, we intend to functionalize the design with additional buoyancy, improving cavities that are hollow and can increase the gastric transit time. Also, it's possible that the push force from the push layer is not sufficient to elute the drug completely and we don't have sufficient drug release. In that case, we would look to change the material properties of the HPM's layer or use a different material altogether that has better swellable characteristics and achieves more efficient drug release. It's also possible that we experience drug entrapment within the pH-sensitive hydrogel cap, in which case we'll look to optimize the thickness of the film and use other fabrication methods like spray coating to make the pH-sensitive hydrogel cap thin such that we can reduce interactions between the drug and the cap and we can have more efficient drug release from the system. Lastly, it's possible that the drug release from the device actually follows a first order kinetics and is dependent on the concentration of the drug left within the reservoir. This is undesirable. In that case, we may look to introduce a constant flow rates control valve into the orificial area to ensure that the drug release is actually independent of the concentration and it follows a zero order release kinetics in a controlled and sustained manner as is intended.